Natalie. I'm one of the senior editors at Methods in Ecology and Evolution. So I think without any further ado, I will hand it over to you, Stefano.
Thank you so much, Natalie, and everybody. Can you hear me well? Someone maybe
Yep, perfect.
thumb up? Okay, perfect. Excellent. So I'm super happy to be here. I think it's the first time that I do one of these live events on the back of a paper. And originally, and the first thing I want to do is to apologize, because originally the plan was to have Eric and Eve, the first author and also the person that led and kind of invented the whole DNA divers initiative. Unfortunately, she's got quite a demanding job, and the times couldn't work. So I'm literally really replacing her for this event, and I promised her that I will make her proud. But I mean, other than just being with this pressure of being as vibrant and creative as Erica, I also really would like to really to credit her for the originality of her thinking and for coming up with this idea that was not originally a plan within her PhD. I mean, if you follow her on Google Scholar, you will realize that she has done a few others different things for her PhD, but also she was really adamant that she wanted to do something that had a more participatory science element to it. And so the first thing that I'm going to do, since this is a live event, before I show you some slides, I'm going to show you exactly what a metaprobe looks like in real life. Sound and all. I will assume that some of you will have already seen it or handled it, but some might not. So it's very light. It's pretty much the size of a maybe baseball ball, slightly bigger than a tennis ball. It has a PLA matrix, so it's a sustainable type of plastic. It's printed in two halves. Obviously, it doesn't quite come out so clean when it comes out of the 3D printer, but then you have to do a little bit of cleanup, and then it just fits precisely like this. Then to hold it together, you need to have zip ties. And on top of that, the zip ties, before you close it, we will procure this little gauze. This is the same type of stuff that you probably put around your finger if you cut yourself. They come in various different brands, and they're quite inexpensive. And what we do essentially, one of these becomes three. We cut once and twice. Now we've got three little replicates in a way, each one secured inside the metaprobe with zip ties, and then the metaprobe itself. Oops, it's closed, and then it goes. Now, how does it go? And here I'm going to share my screen first, and I'll show you quite an evocative picture. Please give me a thumb up if you can see the presentation.
Yeah, that's perfect. Thanks.
Yeah, okay. So that's the metaprobe deployed, as you can see, also with a different echinoderm from the one that was used to advertise this event. And the first disclaimer is that's not how you use a metaprobe. So you don't need to have a nice camera. You don't need to have a nice light that shines over it. And you don't need to have similarly spherical live animals that crawl around it. So this was a little bit of a scenic thing that a diver decided to do, and we're very grateful because it's great advertising material, and actually it's currently also my desktop picture at the moment. So the reality is that we use the metaprobe in a different way. And how was he born? I mean, before we talk about Erica and the DNA Diver initiative, we need to explain why the metaprobe became quite fashionable in our research group, and we hope beyond. It's because, as you can see from my face there, recently, only just a couple of months ago, I was just doing this thing that people working in environmental DNA often must have done, which is to pass water through a filter, and you do that often with a physical, real hand-powered syringe. It can be very, very long. Sometimes it gets stuck. It gets harder and harder. You use a lot of plastic. Even in the situations where you can use a peristaltic pump or a vacuum pump, there is a lot of equipment. You have to carry a lot of stuff around. And the main limitation is that if you say, oh, do you want to engage in this project? Would you like to sample some eDNA? And then the first response is, yeah. And then when you start listing all the measures and all the particular steps that you have to do with a more conventional gold standard eDNA sampling, a lot of times people think, I don't think this is going to be possible. And the first typical example was pioneered by Giulia Maiello, who is also a co-author of this paper. And I must say, like, working in the last few years with Giulia and Erika has been fantastic. They have basically created the synergy where Julia was the fishery person and Erika was using it with the divers, but literally they became the two metaprobe girls, and they together they stimulated each other. They started these incredible collaborations across the world. And as you can see here, on the bottom, you can see what happens to a metaprobe. This particular one is blue. And this is the way the metaprobe first was used in the first place for Julia's PhD a few years before. She wanted to have the fishermen engaged in this. And so she managed to obtain a collaboration with fishermen, and then the metaprobe was inserted into trawl nets. And then it was gathering a lot of biodiversity information. All the way to the right, you can see another way of using the metaprobe when you have like a strong fishing line and a carabiner hook where there is really handily goes through the holes of the metaprobe, and also being heavy metal, it also helps it sink. So that can be used to just dangle from a boat or attached as an arm of a longliner, because then you can use it in association with longlining fishing. But then here comes Erika. Erika decided that, of course, like many of us, she's a recreational diver, and the idea is that, wow, this is a very popular activity, and you have recreational divers potentially everywhere in the world that can go in the water, but also you have also professional divers that have to dive because they have to do some job, okay? And the first thing that Erika did was to start a collaboration with Natural England, which is one of the statutory bodies for nature conservation in the UK, partly associated with the government. And what you can see here to the right, you can see the metaprobe, you probably can spot it between the two tanks. And we conducted this pilot study to show how useful it could have been to have divers that normally survey inshore fishes around the UK equipped with this device, because then when they could come back onto the boat, on the shore, they could just give the sample, the gauze inside the metaprobe for eDNA screening. And this resulted in a report for Natural England, with Natural England, which was really very positive, and it showed that, of course, eDNA is able to collect a lot more data than a diving person that often has to go through very murky water. And that is it felt like it was something that could really change the way we survey fish diversity, and also potentially other taxa around the UK and beyond. But then this had to become a proper scientific paper. And Erika decided that it had to go through a number of steps. And this is just an image of the little package, like after you prepare the metaprobe, like cutting the gauze and tie it, then you end up with a few tubes which contain already the preservative, and the metaprobe is already set up. And so the person that decides to collaborate with this type of activity, they only just have to take some gloves, place it, and then when they finish the dive, they can just place the gauze inside the tubes. So Erika felt that it was very important, first of all, to have a number of divers interested in collaborating as a pilot. So we were talking about just a few divers from Natural England, Sea Search around the UK, And then again, the two key steps before going, expanding the method across the world was to trying to compare the ability of the metaprobe to capture the biodiversity of fishes compared to the gold standard water filtering method. And to do that, she designed the experiment in a way that she could do it in control conditions, and we were able to establish this nice collaboration with the Blue Planet Aquarium in Cheshire. And then, thanks to the volunteers, Sea Search, Natural England, then we could also have divers engaging initially at the Liverpool docks, which is an area where the water can be quite clean, so it's used often by divers to have a place which is safe. The water is not it's not challenging, and people can dive and look around what they can find. And then they decided to also do this test and wear the metaprobe. And then the final step was, can we extend it? Can people go further afield and further away in different oceans, different parts? And so we had a couple of really, really kind and enthusiastic charter boats that were going all the way to Orkney and even all the way to Norway to do recreational diving, and they brought the metaprobe with them. So as you can see from the paper, which actually is in every slide up here, there is a DOI, but you can easily, if you do Eric and Eve and DNA divers methods in ecology and evolution, you will find it, no doubt, and it's on open access. You will see that there are many co-authors, because we decided to offer to our citizen scientists, we could say, participant scuba divers, to be part of the adventure, and the vast majority of them found it nice to be co-author of a scientific paper, and you will see that between the lines, you will probably recognize some of our other group members of the Mariani lab, because they also decided, you know what, I'm going on a holiday. It was a former postdoc in our lab was going to dive in the Red Sea, and she decided, well, actually, I'm going to get you some metaprobe from there. And then another collaborator was going to dive in California and said, okay, I'm going to send you some. And then we had some collaborators from an NGO in South Africa, who also they study sharks, and they said, oh, absolutely, we're going to dive. We dive all the time, so we're very happy to send you some metaprobes. So the first thing I'm going to show you, which is, of course, everything I'm going to show you is in the paper, is that you can see these colors, and these are on the very top right in blue is the Blue Planet Aquarium, and you can see that triangles and circles are all overlap on top of each other. So triangles and circles represent the metaprobe and the Sterivex filters. So you can see that in a very strictly controlled, contained environment like the Blue Planet, basically the community biodiversity is the same. Everything that you capture with the metaprobe is the same that you capture with the filters. But when you go into the other areas, and you have the Liverpool dock and in pinky red, and then two different locations in the south west of England, Cornwall, yellow and green. You can see now that you start seeing the separation within the circles and the triangles, which means that the metaprobes and the Sterivex filters are capturing slightly different communities. And the real difference is that Sterivex were capturing more. So in the first attempt that we did, we just noticed that the metaprobes were not collecting as much, were not able to collect as much DNA and as many species as the traditional water sample. But the interesting thing that we could notice is that the location could still be significantly discriminated with either way, with either method. So that means that if you still have this dilemma, okay? If I have the opportunity and I have all the facilities and I have all the setup and the equipment, etc., to do the traditional water filtering, you probably go with the water filtering because you're going to collect more species. However, if that's not even an option, and the option is zero, nothing, or the metaprobe, then the metaprobe will be able to really this still separate geographical locations around the UK. And now I'm going to show you something interesting about what happens to the metaprobe when it moves and when it doesn't move. And this is another part of another figure of the paper. And you have these three panels, left, center, and right, where you have three different treatments. The one to the left is the traditional syringe and filter water sampling. In the middle, there are metaprobes associated with divers, because the Blue Planet Aquarium, this was all conducted in control condition. Blue Planet Aquarium has divers because they have little dives inside the large tank to show also the visitors of the aquarium how they feed some of the sharks and the rays. And then to the very right, you have metaprobes that were just sunk and soaked. So they were just dangled in a bag in the water without going through the motions of being carried by a diver. And these three little colors, the yellow, the blue, and the black, I mean, the yellow and the blue is not so much of a big deal because they go really together, because the yellow is the molecular operational taxonomical unit, and in blue is the taxa that we decided to accept after filtering and analysis, but they follow the same trend. And in black, you have the subset of species that were elasmobranchs, because we used an elasmobranch specific primers for this particular exercise. And when you look at the elasmobranchs, the first thing you notice is that pretty much the same 10-ish species of elasmobranchs that were stocked in the aquarium in that particular tank are equally identified by the syringe filter and the diver metaprobe. Whereas the soaked metaprobes, and here I'm going to say the 5 different samples you can see refers to different soaking times. So you've got 10 minutes, the first one, 30 minutes, the second one, 1 hour, the third one, and so on up until I think 200 hours. And you can see that at the beginning, you have to go beyond the half an hour. You have to get to at least 1 hour to start reaching the capturing of all the species of elasmobranchs. And it's even more sharp with all the species of fish. And you can see that soaking time makes a huge difference. And with the divers, there is no relationship anymore between the time of soaking and the amount of species you're able to recover, because this is the trick. The divers ram through the water. And this is the greatness of DNA divers. And we can always say the diver maximizes and speeds up the process of accumulation of what we called a quasi-passive filter, because the metaprobe is passive if you let it there, if you let it dangle and soak, doing nothing. But if you attach it to a metaprobe, or if you attach it to a diver, all of a sudden, would you really call it a passive filter if the diver is actually swimming through the water and actively letting the gauze getting completely, getting a flow of water against it, adding as much particulate that is dissolved in the water. And that's quite nice. The bottom graph, I would just don't worry too much about the two, the one on the left and the one in the middle, which shows this pretty much similar behavior of the divers and the syringe. You can see the first column of the right set, which is the soaked metaprobe. You can see that at the beginning, in white, the first column shows that the vast majority of the DNA is human. And this is the metaprobe that has been soaked only for 10 minutes. Because at the beginning, if you keep the metaprobe only for a short time, most of the DNA that you detect is the DNA of the noise of the DNA of humans having to deal with the metaprobe, because as much as you are careful and as much as you wear gloves, etc., you will always, add your own DNA to the operations. But then as you let it soak and you give it time all of a sudden the human DNA will become negligible compared to the large amount of DNA that gets accumulated. And this is another way of representing the impact of the diving action on the ability to recover species. On the y-axis, you have the number of species recovered, 20, 40, 60. And you can see that those two slopes refer to two different tanks. The blue and the yellow, they are two different tanks. And when you and you can see that there are two different slopes, one that is continuous and one that is dashed. Okay, so you can see that for both tanks, you have uh, a slope that the dashed line has less of a slope. So it's less reliant on time of soaking. And that is why, because the continuous line is the slope associated with just the soaking metaprobe. And the dashed line includes the impact of the data point coming from the divers metaprobes, which are here around 50 minutes and 60 minutes, because this was the time of those two dives. And essentially, you can see the data points bringing it up and showing that even for shorter times, that is maybe less than 1 hour, the divers are able to collect that good amount, that good extra amount of species that makes it less reliant on soaking time. And here, Julia appears again. Why do we let Julia appear again? Because, as we said at the beginning, that, oh, look at that, unfortunately, the metaprobe will never capture the same amount. The diver metaprobes is never going to catch the same amount of species as a comparable Sterevex filter. But we had the opportunity to improve the activity, and we focused on, for example, divers that were able to spend more time in contact with the bottom. So working divers that were actually counting benthic species, lifting cobbles from the substrate, looking at the kelp forest. This is a data set from Julia's work along the California coast. And here we can see that over 2 consecutive years, you can see on the x-axis, we have 2023 and 2024 for 3 different sets of sample, the yellow orangey ones, the greeny ones, and the bluey ones. The bluey ones are water filtered in the traditional way in both years. The green ones are now the metaprobe associated with divers. And you can see being on the picture above that is being applied to the divers. Now you have an average number of taxa that now are becoming even more than the water samples. This is because if you let the diver really work and being active in the water, then you maximize the more DNA coming in contact with the gauze. And then you have the BAM. This BAM in yellow orangey to the left is basically the operator is attaching a metaprobe to a fishing rod, and instead of letting it dangle passively, the metaprobe is cast and it sinks to the bottom, and then it gets retrieved, and then it's cast again. It stays on the bottom for 2 minutes, and then it gets retrieved and it cast again. And we decided that 10 times, and every time the metaprobe would go in onto the bottom for 2 minutes, and then we would travel back with the fishing rod. That managed to create sufficient stream through the metaprobe that now all of a sudden you can see that the boat associated yellow orangey data set is very, very comparable to the water. So at this point in time, I feel quite confident that I can say that actually we don't have to be a little bit of apologetic and disappointed, because after all, the metaprobe will never capture as many species as the water sample, because actually this is probably not true anymore. Sorry. I'm going to show you one final picture of the power of Erika's DNA divers idea. And this is an ordination using a canonical correspondence analysis that you can see also the main variables that are responsible for separating this data point. And as you can see, the biogeography is the most important determinant, because you've got 3 main clouds. The very top, you have the temperate Pacific. The bottom left, you have 2 separate Indian Ocean locations, one in South Africa on the side of basically the Indian Ocean side of South Africa, and the other one is in Jordan, so Red Sea. associated biogeographically with the Indian Ocean. And then you've got the big clump to the bottom right, which are all the Northeast Atlantic samples, Great Britain, both southwest of England and Scotland, as well as Norway, and even a sample from Cape Verde that was contributed by one of our team members, Peter Schoen. So as you can see, the diversity of species was staggering, because these are just 39 metaprobe samples coming from volunteer divers that went into this different disparate parts of the world. And out of 39 samples, we collected 275 species. This is, to me, doesn't look like a method that doesn't work. It's absolutely extraordinary, and it does reflect the very different biogeography and obviously well divergent communities that you find in these three different parts of oceans around the planet. One interesting aspect is that you can see here mola mola, the ocean sunfish. It's the only crazy organism, because all the other species of fish you can see in little black dots, they cluster alongside their samples because they are associated with a certain biogeography. The ocean sunfish is a cosmopolitan species, so it's not associated specifically with any one ocean, but it's kind of drifts floating between the Atlantic and the Pacific, between, obviously not in the air. And then, just to give you a measure of how much this 275 species is with 39 metaprobe, I'm comparing it with a very recently published study that actually uses samples collected globally across the 5 continents through a large network that collated different samples from, again, temperate, polar, tropical, Atlantic, Southern Ocean, Pacific, Indian Ocean. And with 936 samples, they essentially obtain 1,225 species, which is a large number for sure, and more than you would ever be able to census with your underwater visual sensors, for example, if you were a diver. But the interesting thing is that the sampling effort in this particular case is like more than 20 times, 25, 30 times big as ours, but the number of species recovered is probably only 4 times. So that shows you how incredibly you have a potential for an army of citizen scientists across the world that are willing to just do what they love doing, and in the process of that, providing regular, powerful biodiversity assessment data. And with the final slide, which obviously portrays Erica with some of the volunteers at the Liverpool Albert Dock, I would like to say that this thing is actually working, because very recently we also published another study that was based on recreational divers from Seychelles that contributed samples. And again, we detected about 160 species of fish with about 40 metaprobes, and we could detect differences between protected and unprotected areas, from coral reef sites and rocky shore sites. And now we have a large network of extreme divers that dive into marine caves that have decided to dive with metaprobes. And into the new year, they will contribute these metaprobes to find out what they discover. And from the divers, we are now also seeing that people that use other types of recreational activities, such as paddleboarding or kayaking or windsurfing, that really want to attach the metaprobe while they do what they do, and because they're curious and they want to know what's in their sea, what's in the areas that they love spending time at, what they harbor. And at this point in time, I think the very important aspect of this type of initiative is that we leave time for questions and conversations, informal as possible. And so I'll stop it here. Please let me know if you want to go back to live face or the slides. We can do both. I mean, I have them close to me.
Cool. Thank you so much. Seems like such an amazingly useful tool and yeah, great stuff. Does anyone have any questions? So like I said before, feel free to just type in the chat or unmute yourself and shout out. I mean, I've got a question whilst we're waiting, which is I think you kind of already covered this a little bit at the end, but what's the next steps with this? So what's the next project that you're planning on using this tool for?
Yeah, so I think there are two main levels. I think I would say probably three because there is an operational one. Okay, so the first two are basically really, I mean, we call it DNA divers. We published this paper which is basically announcing it. And we had another couple of examples where it was nice to apply, but it's not really consistently regularly happening, right? It's we really want it to be used because something that I didn't say, it's an incredibly affordable way of doing things because most people now have access to a 3D printer. This not only is this cheap compared to, if you use a static filter and a syringe, you already have, every time you have to use 10 pounds, and every time you have to buy a new one because then you have to dispose of it with a lot of plastic waste. This thing goes into a mild bath of bleach and you rinse it and you can use it hundreds of times. We never broke one. It's very robust and it actually has spent time at more than 70 atmosphere in commercial trawl nets in the Mediterranean, in the midst of dozens of kilos of fish being fished. And it's incredibly robust and so flexible, so versatile that I think that it's got a huge potential. So we really want to make use of it. So we are engaging, and I mentioned at the end of the presentation that we now are attracting engagement. A dream that I have is that one day there will be some diving providers that can also offer, hey, apart from just taking you to this beautiful coral reef, would you like to wear one of these? Because we've got a collaboration with someone who will be able to find what the DNA tells us. So this could become potentially even commercial. Now, I don't want to make money out of it, but it's possible that maybe some people that actually organizes these things could create a revenue if a person is interested to, I don't know, if on top of your diving trip deal, if you add maybe a few 50 quid extra for a week, it might give you a lot of samples of DNA and biodiversity uncovered. Final thing I wanted to say is I would like to be braver in the sense that up to this point, when doing research on environmental DNA quite regularly, we have to sample certain locations and we say, okay, we have to sample 500 samples over a certain number of seasons. What do we do? Do we still buy all these filters and we get the tubing and we get all the bottles? Or we just do away and use the metaprobe now that we have so many examples of how we can use it? And I'm still 50-50 partly because you worry that, when you go and publish, there's going to be some reviewers that says, no, this is not an official gold standard method, so you cannot do it. Or you should have done both and spend twice as much because you have to compare. And how long do we have to compare? So I would like this type of studies to make me bolder and say, you know what, next time I'm not going to buy the filters. I'm just going to do it this way. I'm going to use this.
Yep, no, that's a really good point. And yeah, it's amazing that they are so resilient and you can reuse them. That's really cool. I have another question for you. So Nimna says, "Thank you. That was really interesting. I'm curious how you account for the fact that eDNA can be transported from somewhere else. So when using metaprobes with DNA divers, how do you tell if a signal is truly reflecting local biodiversity?" Or if it's from, could be from eDNA that's drifted in from other areas or persisting from a long time ago.
Yeah, so an excellent question, and I would argue my answer, I guess, goes to the general features of eDNA. This is something that could easily apply to any other type of eDNA sample. It's if you are sampling with bottles and filters, and then you pump the water through the filter, then you will also wonder, oh, where is the DNA that I collected from this water bottle, whether it is a hand maneuvered bottle or whether it is a Niskin bottle, that also is exposed. Whatever volume you capture is exposed to oceanic currents, and that means that inevitably you have to take into account some transport. Now there are a few papers now that use the information about the prevailing currents of the area where the operators have been sampling to have a bit of a probabilistic estimation of the radius from which the DNA might have come from. And I think that this is very legitimate, but I do not think that the metaprobe is particularly disadvantaged, because in a way the metaprobe, if you dive in a certain area, you are less likely to stumble into one bottle that accidentally had an overrepresentation of certain things over others, because you dive for 40 minutes in a certain area, so you're trying kind of sweeping everything that is in there. But there is also a drawback, and I think that the metaprobe's drawback is very obvious, because when you drop this guy into the water, then suddenly here it is. From the moment it goes into the water, potentially it starts collecting DNA. So a diver that wants to only measure stuff that it's on the bottom at 50 meters, then obviously in the moment they go down, they're beginning to accumulate. But obviously, in the first few minutes, you don't accumulate too much. You could also put it in a plastic bag and then only open it and attach it, maybe with a hook at the depth that you're interested. Or if you're using a deployment from the boat with a rope or with a line, again, the same issue. You might decide that you're going at a certain depth, but the metaprobe will obviously, when it comes back to the surface, is also capturing and raking DNA also in the water column all the way to the surface.
Cool. Sorry, I keep turning my camera off because I've got a cold and I don't not like everybody see me sneezing everywhere. Okay, Martin has another question. So he says, great presentation, interesting technique. Is it possible that the professional diver and the angling retrieved metaprobes would record a higher number of species versus the static water filter samples simply because they were taken closer to the sediment? So divers picking up boulders, angling, casting metaprobes, resting for 2 minutes on the seabed, and therefore you would expect more demersal species to be recorded.
What can I say? The answer is yes. So we actually, I didn't show it to you, but from the data that show that extra little bonus of diversity in the diver metaprobes in the California project compared to the other methods, we also coded the species that were detected, and we have a higher percentage of very benthic species in the divers activity compared to the water, because obviously we have also some species that are very, very happy pelagic that the divers with the metaprobes going on the bottom missed, but there is a higher percentage of species that are actually only found in the metaprobe, and often they are that many of these are really, really benthic, and some of them are really, really cryptic that only live between crevices, between stones. So absolutely yes, that's exactly what happens.
Cool. And then one more question from Sumit, who says, if I'm understanding correctly, the probe needs to get close to the species to collect enough eDNA. So what's the minimum bound for eDNA to be detectable, and what scale is used for quantity of DNA?
Um, uh, i- if I understand well, did the first part was about getting close to the actual animal? Yeah. So no, not necessarily. I mean, I suspect that you are diving, if you are diving and there is a seal that follows you, or a dolphin, or, hopefully not a great white shark, then you probably are going to detect a lot of that DNA of that individual animal that is spending a lot of time close to you. But in general, uh, eDNA, the assumption of eDNA is that you are not requiring the proximity of an individual animals, because you are collecting the samples from your samples are basically shed DNA, which is either free with particular organic matter or in cells that have been already shed because the scales have fell off the fish, or it's in the poo of the fish or whatever. So there is no assumption that we have to be close to animals, although there are obviously studies that that compare the probability of detecting a certain organisms based on how close, on how recent it was. In terms of uh, how recent the signal is, we know it pretty well. We know that most of the DNA in an average marine water gets degraded within 24 to 48 hours. So it has to be fairly fresh, but it can last a day or two. So we know that if we detect that organisms, it was there no further away in time than a couple of days ago.
Cool. You're getting a lot of questions today. So another question from Ivan is the gauze inside the probe standardized? And does it need to be for you to compare between studies?
Very good point. So we tend to use a fairly standard medical gauze, which has a I think I don't remember. I think that the mesh is 0.5 millimeter. So if I stretch it like this, you can see that 0.5 millimeter is very little. So in theory, if it was just one panel, most of the DNA would go through, right? Unless there are larger chunks of things. But this is rolled, and this is rolled 50 to 100 times. Okay, so if I did this, it's 50 to 100 layers. So when it's so packed, those three things act like three sponges essentially, and they are little sponges, and they soak and soak and soak. At some point, when in certain areas, when you take it out, it's a bit brownish because you can see that a lot of stuff has accumulated. But this is a very good question because it raises the point that quite a few other people in the past asked me, "Hey, have you tried this other material? But have you tried this other material? This other material should have DNA stick more." And I have to admit, and some of my more junior brilliant researchers that have been working with me in the last few years have proposed maybe we should try to use different materials in the metaprobe to see which material is absolutely best. But I was just too exciting to just sample more stuff from more places. And since this is working pretty fine, we keep using the pretty standard medical gauze. And the jury's out. If some other group has the time and passion to use different materials and test the metaprobes and to see what is the golden material that traps the best DNA, then I'd be absolutely super delighted to learn.
There you
And I can't
go.
promise I'm going to do it myself.
A free paper idea there for
Mm.
people. Okay, so I think final question then is from Mia. It says these are cool and valuable results. If this initiative eventually expands into a full-on community science project, I'm sure the divers that do data collection will be really curious about their findings. Do you have any ideas on how you're going to communicate those results back to the citizen scientists?
What a great question this one is as well. So we feel a bit. um, not guilty, but we haven't done enough. That's for sure. We prioritize the paper. We prioritize the report for Natural England. We prioritized Erica's PhD thesis. And Erica herself, during the PhD, she said that she was planning to actually have a little website, maybe part of her GitHub page, where actually people can click and see a map and click on the location that they contributed to and see on that dot that you click and then the list of species comes out. That is ultimately, I think, something that could be really nice to have, an updatable, open access, open source little map with a list. And I think that at this point, so obviously, Erica communicated with every contributor and provided a list on a personal basis by email. But I think that if something like this becomes established and very popular, I think it would be really important to have something like this, like an openly accessible map where everyone can click on a dot and see what species on what date the metaprobes were able to collect.
Yeah, that sounds really cool. That would be amazing. So I think that's probably all we've got time for today. I think we've run out of questions, but thank you everybody for asking questions, and thank you again, Stefano, for coming along and talking to us. That sounds like such a useful tool, and it sounds like a lot of people are hoping to use it in the future.
I hope so. I hope so. Please do have a go and get in touch. Of course, anyone who wants to have a little bit more um, guidance and share, I can happy to share experience.